This is the title of my talk, Tiny Lights, Big Impact, How Luminescent Nanoparticles Revolutionizing Medicine or Nanomedicine. I'm going to take you into a journey of my scientific career. It started somewhere in the 1990s where I was working on fiber optic sensors for chemical contaminants. They were beautiful, elegant, but they were large, something like 0.5 millimeter. So large in the context of working with cells and tissues, biological cells and tissues. So this has coincided with the emergence of nanotechnology as a field. And we move slowly but surely from 0.5 millimeter fiber optic sensors to microparticles. Those are micron-sized particles, 1/1,000,000th of a meter. And then from there to the nanoscale, which is 1/1,000,000th of a meter, about 100 folds smaller than the diameter of a single hair. Okay? It started for me as some sort of an accident. I read a couple of interesting papers in the journal Science, which is the leading scientific journal in the world. Every Friday I was reading this journal. And I read two papers, back-to-back papers by two friends of mine. One of them named Shu Ming and the other one named Shimon, back in Emory and UCLA, showing how you can take those colloidal, inorganic nanocrystals named quantum dots that we knew about them from previously, where they were work was in organic solvents, not in water. And they showed in those two back-to-back papers that you can actually transfer them to aqua solution, to water solution, or biological solutions. And that got me dancing in the office. I called one of my students and asked her to actually go to the lab and synthesize those particles the way it was described in the papers. And she did. She came back 3 days later with the results, and I was astonished. Her results were better than the ones reported in the paper. So immediately I called all the students and moved the entire lab to quantum dot research. Okay? 2 months later, we discovered that her results were a mistake. It was too late to change. This is how I started my career on quantum dots. What are they? A quantum dot is a crystal. It's very small, a few nanometers in diameter. It's so small that it's effectively, we call it a dot, hence the name quantum dot. They are made of semiconductor materials like silicon. We often use cadmium sulfide or cadmium-containing materials, which is a problem because cadmium is toxic. We'll talk about it later. And there is some feature in this material which called the energy band gap. It's an energy difference between two bands or two energy levels in a material. In a bulk material, it's constant. So if you take cadmium sulfide material and shine UV light on it, you will get some yellow, faint yellow emission, not much. But when you go down to the nanoscale, something magic happens. This band gap of energy difference between the energy level is actually changing. As you decrease the particle size, it gets bigger. So the color of the material, the color of light emitted from the material is changing as well. The material doesn't change, just the size of the material change. It moved from 1 nanometer to 10 nanometer. That's all, very small change. But that actually results in a tremendous spectrum of colors. And we decided to actually making them and use them in various applications. And today I'm going to talk mostly about nanomedicine because of the interest of the crowd. But there are other applications. You can see here the first demonstration of using quantum dots in cellular imaging on the lower left side. Only two types of quantum dots here with two different colors, but we showed over the years that we can actually tag cells with 6 or 8 colors of quantum dots for 8 different markers. Now, this is not a simple particle. I can present it as simple, but it's not. Actually, in addition to the core materials, there's a passivating shell, and in addition to the passivating shell, there is a protective coating, sometimes a polymer coating, and there are many molecules like antibodies that can recognize specific cells or drug molecules that can actually treat tumors effectively, locally, in a targeted way. And all of it requires very controlled, precise elegant chemistry. And this is where the lab is busy with the work of many dedicated students that are actually perfecting the structure, the architecture of these nano platforms. So one example of this technology that you all have either in your hand or in your pocket is a cell phone. The cell phone display is actually contain nanomaterials like quantum dots. In the early day of light-emitting displays, we use what we call LCD, liquid crystal displays. And in there, you see there was a white light source, and each pixel on the chip was coated with 3 different colored filters, which was a difficult process to do. Okay? And also, there was a lot of noise, and it wasn't clear. In the quantum dots screens, what you have is a blue light source and a mixture of red-emitting and green-emitting quantum dots to generate 3 different colors, blue, green, and red. The result of this is something like that, a display that will have billions of pixels with distinct, pure, different colors. And you can see the difference in how fall colors this time of the year look like in the conventional television on the left and in a Samsung QLED or quantum dots television on the right. The difference is very clear in the color and also the visualization. So now we take it to medicine and we use it to visualize cells. So on the left, you see a single cell decorated with those red-emitting quantum dots. Impressive, but less important. On the right, you actually see results of 3 biopsies of breast cancer tissue. Okay? And you can see that each column here will have 5 different markers, specific markers. Each of them is tagged by a different quantum dot. And you see a difference, a clear difference between biopsy 1, biopsy 2, and biopsy 3. That allows clinicians to design individualized, targeted treatment for breast cancer in contrast to what it was used before. Everyone would get the same treatment. Okay? That minimizes side effects and increases dramatically the prognostic outcomes. Okay? So when you move, it's not only working in culture or pathology culture, it works in whole animals as well. We can actually embed or target those quantum dots to tumors in a whole animal. And you can see them with your naked eye. So it's not something that is requiring sophisticated equipment or something like that. Okay, so in addition to imaging, we can use those quantum dots as a form of treatment. In one treatment for cancer is named photodynamic therapy. In photodynamic therapy, the goal is to produce reactive oxygen species that, like you see those oxygen or O2 species there, that will attack tumor cells and kill them, basically. And it seems that quantum dots are doing it very effectively, as you can see on the upper right. That's a lot less painful than surgery, a lot less painful than showering yourself with liquid nitrogen, as it's often done in dermatology clinics. Okay, so none of this is possible without hard work, and there's more to do. We need to, it's an ongoing work. We need to design better particles. We need to develop detection platforms to visualize them better, and we have to make them less painful and more targeted therapies, and alleviate this dogging issues of nanoparticle toxicity that is still a remaining very difficult challenge. So a lot of people were involved in this work, my students, current students, and former students, overall 25 PhDs over the years, and counting, 10 master degrees, 10 postdoctoral fellows, dozens of collaborators in the US and around the world in 4 different continents. Lots of work, funding agencies that supported this work. And I don't want to forget, as a last minute, some things that we often forget, that all of this work cannot be done without the infrastructure that we have here at the university. And the infrastructure is not only equipment and buildings, it's people. The labs don't clean themselves. The water doesn't flow without maintenance. There's no electricity if someone doesn't care of it. And honestly, in a chemistry building, We need to keep this chemistry building very safe, because if not for this environmental safety people that come and inspect us and make sure that the building is safe, this building could have caught fire right now and even blow up. So we have to remember them and thank them from the bottom of my heart. Thank you very much, guys.